You've got a lot of information that just about every hallmark of aging in mole rats is abrogated and delayed. So their telomeres do shorten as they get older, but they reach a certain level and that activates telomerase, and then they maintain that level for prolonged periods of time. So in a study in cell culture, most cells for mice get to about 18 to 20 population doublings, and then the cells stop proliferating. Hayflick limit for humans was 52. We've got to 160 in the naked mole rats, and that took multiple years, nearly 8 years worth of growing cells in culture, and we don't see that senescence phenotype. And we think it's because they can maintain their telomeres and telomerase. With regard to proteostasis, every aspect of proteostasis seems to be tightly regulated and well maintained as the animals get older. Their protein formation is much slower than what you see in mice, and their protein half-lives seem much longer. So their proteins seem much more stable with lower rates of synthesis, so there's less error or more time to correct error when they're making new proteins. They seem to have very high levels of heat shock proteins or molecular chaperones, which I think contribute to the fact that their proteostasis is so well maintained. They're able to maintain their protein structure more readily. There's high levels of ubiquitin, but the proteasome, the garbage disposer of the cell, seems to be able to work much more efficiently than that of mice. And even autophagy, which gets rid of protein aggregates and mitochondria and things like that, when we do it in culture, the maximum rate is much, much higher than mice. So they can ramp up their machinery quite efficiently to get rid of damaged proteins and well maintain proteostasis. I think that's a very important component of their extreme longevity.
Today we're joined by Dr. Rochelle Buffenstein, a comparative biologist and research professor at University of Illinois, Chicago. She has authored over 200 scientific publications, is a fellow of the Gerontological Society of America, and a former president of the American Aging Association. Not to mention, of course, that she's the world's top expert on naked mole rats, the bizarre and wonderful long-lived rodents that seem to defy aging. Rochelle's groundbreaking research has shown these animals don't just live unusually long, they resist cancer, stay fertile, and avoid the typical rise in mortality with age. Rochelle has spent decades unlocking their secrets, and today we'll explore what makes naked mole rats such a powerful model for understanding aging and what it might mean for us. Rochelle, welcome to LEVITY.
Thank you very much for the invitation.
I've thought for some time that if LEVITY had a mascot, it would be the naked mole rat. And after this episode, I think people will understand why, if they don't already. But maybe we should start with you and your background. What drew you to biology, and how did you end up working on naked mole rats?
Yeah, that's a question I get asked often. I am from Zimbabwe, from Rhodesia, hence my accent, in case you wondered. I grew up on a farm there, surrounded by many interesting animals, and from a very early age, I had a deep fascination with every animal that came about. I think to my mother's chagrin, I was always sort of keeping creepy crawlies and chickens and anything injured. If I found an animal, I was interested in understanding if it was going to survive and what I could do to help it. But my interest in naked mole rats is much more serendipitous. As a student coming from Rhodesia to South Africa to study, we had very strict exchange control, and I needed to get a job as pretty soon after I arrived. And I managed to get 2 jobs in the zoology department. I didn't want to work as a waitress or in a bar or anything like that. I wanted to do science. And one of my jobs was feeding all the critters that the scientists at the University of Cape Town had there, which ranged from bullfrogs to goats and naked mole rats, of course. And my other job was working as a research assistant to Professor Jenny Jarvis, who had moved from Kenya with her colony of naked mole rats. And so my job was to feed them and to do whatever things were needed for her research. But one of her problems was that she could never get the naked mole rats that she had brought with her to breed. She kept them in her office, so it's a long way away from animal facilities and all the new rules that go with that. And her research was more of an anatomical nature. But she was at a conference with Dick Alexander, a Eusocial insect biologist, who spoke about why you would never find eusociality in a mammal. And as he was talking, he was explaining the only hypothetical scenario in which this kind of situation was possible. And the man sitting next to Jenny, Terry Vaughan, another pretty famous zoologist in those days, turned around to her and said, "Hey, he's describing your animals." And so the penny dropped. The more they thought about it, it was very clear. Everything made sense in the light of Dick Alexander's hypothesis. And Jenny applied for a grant from National Geographic that I helped her write and did all the figures for, in effect. And we went to Kenya 6 months later to collect the naked mole rats. And even though I've worked on many, many species of animals, I always come back because they're such interesting critters in so many different ways.
Maybe we should go right into it then. What is a naked mole rat? What do they look like? Where do they live? How do they organize socially? You've alluded to it already, but...
Okay, so a naked mole rat is a very good looking, as I'm sure you'll see with all the different pictures, mouse-sized rodent. It has a big flat head and large protruding incisors that make them look more like a sausage with teeth or a baby walrus, if you're using polite descriptions. The natural species range is in the Horn of Africa, sub-Saharan Africa. It includes countries like Djibouti, Somalia, Ethiopia, and Kenya. And here they live a strictly subterranean existence, living in an extensive maze of underground burrows that may extend as much as 3 kilometers in one direction and 2 meters deep. They seal the burrows off so predators can't normally enter them except in the rainy season when they're expanding the burrow system. They live in large family groups, which I think helps them, to a large extent, find the patchily distributed food in their burrow system. Once they find appropriate food sources, they very carefully don't eat the skin. They peel into the burrow, into the tube, I should say, and excavate the interior, eat the storage components of the plant, and then stuff it with soil so as to sort of not kill the plant and sustain a readily supply of nutrients going forward. And I would imagine that the outer layers of those plants are pretty toxic. That's where the secondary defense mechanisms of plants are. So they've evolved mechanisms to not have to deal with those poisonous components of plant material.
Is that also why the teeth are kind of outside their lips, so to speak?
Yeah, they dig their burrows with their teeth.
Yeah.
Using powerful jaw-like actions. So if you think of those teeth as chisels, 25% of their muscle mass is in their cheeks, and they form very powerful chisels, and the teeth get worn down. So when we're capturing them, if we don't capture them the first day or so, eventually we find animals with very short little incisors that they've used for digging. And another adaptation is their lips close vertically. So they've got these hairs on their lips and their lips close vertically so that they can dig without getting soil into their mouth.
Right.
So it's an interesting adaptation to life underground, and they've lived underground for at least 18 million years, and have evolved an incredible suite of adaptations that enable them to thrive in a very hostile environment.
Yeah, and as you said, they are eusocial animals. What does that mean?
So eusociality sort of is the ultimate of social. Eu meaning good or bad, euthermic, good thermoregulators. So what that means is they lead a societal life very similar to what you see in the bees and ants and wasps, where reproduction is monopolized by a single breeding female, the queen, so to speak. And then the other animals form tasks of burrow maintenance, food collection. They bring the food back to the nest, they extend the burrows, and they maintain the colony. So there's a very pronounced reproductive skew. Only one female breeds in the colony, and she will mate in captivity with the male I pair her with for life, but she'll also take on 3 or 4 of her sons as the colony gets bigger. So there are very few reproductive animals. Less than 1% of the animals ever get a chance in their lifetime to breed. And that's the reproductive skew that defines eusociality in mammals.
How do they survive these extreme conditions underground? There's not a lot of oxygen, or maybe nutrition around, and it's very dark.
Yes, if you think about them living in colonies of up to 300 animals, they rest together in deep nests the size of a football. And you would imagine that in the nests especially, there's very little oxygen to go around. There's a lot of carbon dioxide present, and the air is very humid. So they've got to contend with hypoxia, hypercapnia, and high humidities, which impair metabolism, heat exchange, and just the getting rid of the waste products is totally dependent on the soil porosity because it's all through diffusion through soil. So those are conditions that they have over many years, millions of years, managed to figure out how to cope with. So they're very tolerant of hypoxia. I've kept them in the lab at 5% oxygen for a week, and then my oxygen cylinder would run out, and then there'd be a splurge of oxygen, so I'd stop the experiment at that. But I would imagine they would continue surviving pretty well under very low oxygen for prolonged periods. Tom Park has shown they can survive 18 minutes without any oxygen in a pure nitrogen atmosphere. And even when you take them out at that time, you'll find that although they're unconscious, their hearts are still pumping and they're breathing at a very much more lower level. They're still alive. If you take them out, then they recover completely with no signs of brain damage or any other change in physiological function. With regard to carbon dioxide, you and I know that when you're in a high carbon dioxide or high ammonia environment, your eyes start to burn and you start getting teary and things like that. They don't show that response. They seem to be insensitive to acid, which is carbon dioxide and water makes weak carbonic acid, in their face and in their bodies. And we think that's due to the fact that they lack substance P, which is a potent pain receptor pathway that activates the sodium trip channels, which are then switched off completely so that they don't actually respond to high levels of carbon dioxide. They have many mutations that seem to be linked to tolerance of high carbon dioxide. In fact, if you look at their lung function, when we put mice or humans, but let's just stick with mice, into environments of high carbon dioxide, mice die pretty quickly. And what it is that kills them is that their lungs fill with water, they get edematous and they drown. And that is a feature that's very common in all mammals, and yet it's not seen in mole rats. We've taken mole rats to 50% carbon dioxide environments and measured the amount of water present in their lung, and it's no different to that when there's no carbon dioxide present. So they've learned to live with this high level of carbon dioxide. In fact, they seem to need a high level of carbon dioxide for normal function. And in situations where they have very low carbon dioxide, they seem to go into some form of convulsion. That was work that was recently published by Michael Zion.
And their ability to survive for long periods of time without oxygen, what's the explanation for that?
So that's an interesting story. Tom Park suggested that they run purely on glycolysis, so the anaerobic metabolic pathway to provide their ATP. And in most mammals, glycolysis runs until lactic acid builds up, and then there's a negative feedback loop that switches off glycolysis, and you die because you can't make the bare minimum amount of ATP. In the case of naked mole rats, it seems that the negative feedback from lactic acid to switch off glycolysis is overridden. Instead of going through the normal glycolytic pathway, they shunt to using fructose as their main fuel. And that bypasses the enzyme that is responsible for switching glycolysis off when lactic acid accumulates. So he has published that, but there's other mechanisms that are clearly evident as well. They seem to retain very high levels of glycogen in their tissues, especially in their heart, which enables the heart to maintain function even at a very low rate for long periods of time. So what I didn't mention is even when the animals appear to be dead at 18 minutes, if you open them up, their heart is still pumping very slowly, but it's still pumping. And we think that this mechanism of having additional glycogen stores plays a role. Their heart also retains many fetal-like features. Actually, the whole animal does, but when we're talking about the heart, the different kinds of proteins involved in contraction, the myosin proteins, are the same as what you see in normal mammals in the fetus, because the fetal environment is also hypoxic, if you think about it. The fetus isn't getting 20% oxygen, it's getting whatever oxygen is flowing in the mother's blood, which is more like 3 to 5% oxygen. And they seem to retain right throughout adulthood these fetal proteins, which enable them to function in a low oxygen environment.
Yeah, it's so fascinating, and we have not even yet gotten to the good stuff.
Going back to the issue of reproduction, you said that very few of the animals get to reproduce. What is it that decides who get to reproduce? And also, is there a lot of inbreeding and problems with inbreeding in colonies like that?
Two good questions. So any female in the colony is capable of breeding. And should the breeding female die, or should a female be separated from the presence of the breeding female, they will be able to become a breeder. And that can occur at any age. So an animal as young as 6 months can become a breeder if isolated from the queen. And an animal as old as 25 became a breeder for the first time in my colony. So what happens when the breeding female dies is that all the females, or a cohort of females, I should say, fight to death to become the dominant female. So we lose a lot of females when a breeding female dies, unless we get in quickly and separate those that are fighting, because those are more likely to become the next dominant queen. And it seems that the breeding female suppresses reproduction in all the females such that they become anovulatory, primarily through bullying and aggressive behavior rather than through pheromones. I can't remember your second question.
The second question, but this here was really fascinating. I mean, it's such an unusual. Are there any comparable mammals in terms of reproduction?
Well, you see in baboons and primates that there's a dominance hierarchy, and the dominant alpha baboon will get the first choice of mates and most likely be the most successful breeder. And should her pup, or what do you call a baby baboon, die, she will often grab the infant from another female and either kill it or nurse it. So you do see dominant behavior in mongoose and in primates and things like that, but nothing like what you see in the mole rat family. So if you look at naked mole rats, the only other species that shows a similar kind of eusociality is the Damaraland mole rat, which also has this reproductive skew with only a few animals breeding, but their colonies are much, much smaller. A naked mole rat colony gets up to about 300 individuals, whereas the Damaraland colony only gets to about 40. And she maintains that reproductive suppression for many, many years. Most animals never leave the colony of their birth. They stay there for life and help maintain this one structure. You asked about genetic diversity, and in the early studies, the hypothesis was that there'd be so much inbreeding and the colonies are so isolated that you would see a lot of signs of inbreeding. But ironically, we're about to publish a paper that shows that's absolutely not true. Most of our colonies were collected in 1980, and because the breeding female stays in charge of the colony for many years on end, we're not up to thousands of generations of new colonies. We're only probably 5 or 6 generations away from the field caught animals in terms of the bulk of our animals. And we find they have the same amount of heterozygosity as what you see in humans, which surprised me no end. We also found that colonies from Ethiopia separated by rivers or mountains were more similar to their southern Kenyan relatives than they were to each other. So we're seeing that there is some kind of geographic barrier that might be playing a role in speciation, but that for the most part, the Kenyan and the Ethiopian animals are very, very similar, with very little genetic diversity in terms of globally, but within the colony, there's still quite a lot of genetic diversity.
So we're talking about eusociality, but there's also these mortal combats concerning the privilege of being the queen. It's fascinating. I mean, how do they kill each other?
They are vicious. Those incisors that they use for digging, if they just want to establish dominance, they link teeth and have tug of wars and bite around the face. But if they want to kill you, they go straight for the lungs and heart. And so you find that most animals, when they're fighting for dominance, get very badly injured paws and very badly injured areas around the face. And then the animal often doesn't die in the fight unless the dominant one really wants to kill it, but it becomes submissive. It goes and sits in the toilet chamber and will stay there and sort of, I don't want to be too anthropomorphic, but it sort of becomes depressed. It doesn't eat and it just remains in the toilet until it either dies or we remove it. So what I didn't tell you when we were describing the colonies was that in captivity and in the wild, they're very clean animals. They maintain a nest area free from excreta. They have a latrine or toilet in the wild quite close to the nest, but also multiple toilets scattered throughout the colony. If an animal dies, they'll bury it in a toilet and seal it off, so they're not keeping dead, possibly infectious animals near them. And in captivity, their toilet chambers are sort of where most of the dominant structure seems to be most evident. So the queen will do her ablutions in the doorway of the toilet, whereas the less subordinate or riffraff, so to speak, will go right into the toilet and clean the toilet and pile the wet or dirty bedding against the side. So
I mean,
when I say that the female who's lost the battle goes and sits in the toilet. It's a real phenomenon.
I mean, how can you not ascribe rather advanced mental states to these animals?
I do think that they're pretty smart. In fact, we've been trying recently to do a study looking at cognition. We're trying to sort of see as they age, do they show signs of forgetting where their food is or not, or being able to learn new things. But just looking at their basic behavior, they live in a complex maze of underground burrows. They can find their way to the nest. They go back to different food sources that they know are good food sources and carry food back to the nest. And they seem to know their place in their little society. So I'm very interested in trying to find ways to really assess cognition. But so far, they're beating me in this game. They're not sharing their secrets.
Another interesting quirk with naked mole rats is that they are somewhat cold-blooded, no?
I hate that word cold-blooded because I don't think it's a good way of describing them.
How would you describe it?
I think the animals are thermally labile.
Yeah, okay.
And what I mean by thermally labile is they can be very hot-blooded. If you put them in a hot environment, they tolerate body temperatures of 42 degrees centigrade for several hours with no ill effects. And they tolerate body temperatures of 12 degrees centigrade with no ill effects. So they can be thermally labile. But thermal lability isn't a normal phenomena in their thermoregulatory profile. They come from a very insulated underground burrow system in Kenya, in equatorial Africa, which is really about 27 to 30 degrees year-round, and living in social groups where they huddle and can't lose heat through evaporative cooling very effectively. They can maintain body temperature pretty precisely in their natural environment. So if I were to use a Greek term to describe their thermoregulatory profile, I would say they're stenothermic. They can regulate temperature very precisely, but only in a very narrow range. If you take them away from their colony and isolate individuals, their body temperature tracks ambient. So yes, in terms of cold-bloodedness, that would be how you would say that they were cold-blooded. But they do have very large amounts of brown adipose tissue, brown fat, and they do use non-shivering thermogenesis, so they can employ endothermic endogenous mechanisms for maintaining body temperature. And what's intriguing to me is that the brown fat is not located like it is in mice, simply between the two shoulder blades, but it goes all the way up the neck, sort of insulating the carotid, so it keeps the brain temperature very precisely regulated. So
Right.
you would get the feeling that they are able to regulate the important parts of their body temperature pretty precisely. So I think it's a myth to say they're totally cold-blooded. Yes, they are when they're isolated from the colony, and they can tolerate it. Whereas most mammals, if they become hypothermic, unless you warm them up properly, they will die. These guys can breed when their body temperature is 25 degrees centigrade, and they will produce fewer pups in a litter when they're being maintained at a low temperature. And the interbirth interval will extend, much like it does in reptiles housed under cooler conditions. But for the most part, in their natural environment, they would be regulating body temperature pretty precisely.
It just shows this extreme adaptability they have in so many areas. I mean, you have kept one of the largest colonies in captivity for several decades now.
Tell me about it.
Yeah, I want you to tell me about it. What is it like maintaining a lab full of naked mole rats? Is it as strange as it sounds?
I've managed over the last 3 decades of keeping animals in captivity to optimize conditions. I I could tell you how to house them in Rolls-Royce or Four Seasons hotel type facilities, or I can tell you the Motel 6 style that I tend to use, which works pretty well. It's labor intensive, but it's not that difficult once you've sort of got the temperature of the room right, given them an electric blanket so they can always find a warm place if they need to. And the breeding females especially need that electric blanket because they maintain their body temperature at 38 degrees centigrade when they're pregnant. So we give them a thermal blanket. We give them enough cages so they can make a toilet or 2 toilet chambers, and we give them a pantry chamber. So it's not that difficult to house them once you've got that down pat.
How much of a difference is it compared to? I mean, it can't be easy to recreate their natural habitat, right?
It's harder than, you can't keep them in a mouse room. They don't drink water, so you have to give them fresh fruit and vegetables every day. Like humans, they're naturally vitamin C deficient, so you've got to give them foods that are rich in vitamin C. That's the big difference with them and mice. But as I said, once you've got the systems all set up and the cages all set up, just apart from bringing in kilograms of sweet potatoes every day and fruit and vegetables, they're relatively easy to maintain. So if you want a colony, I'm happy to give you one.
Can I ask you, are they considered a pest in Africa? Do they eat the farmers' crops? Or, I mean,
Yes.
if they grow potatoes or...
Yes, so our best collection was in a sweet potato field, and they were very happy that we came and took their pests away. They do eat them, which is quite interesting. When they find their pests in their sweet potato fields, they hunt them, or people hunt them. Their meat is much more like steak than chicken. So mice, if you look at a mouse leg, most of it looks like chicken. It's a white flesh. The mole rat flesh is dark red, so it seems to be predominantly mixed fiber, anaerobic, slow twitch. They don't hop and jump like mice do. They walk on all fours more slowly and rely more on aerobic muscles.
So the farmers, when they catch them, they grill them and eat them with bread, or...
I don't know if they eat them with bread. They probably just
Little olive oil.
I wonder, have you tried them?
No. I haven't brought myself to that, although Tom Park, whom I'm working with now, keeps threatening to put them on the barbecue. We haven't done that.
Yeah, but is it expensive though compared to housing regular like black six mice that everyone uses?
It is expensive to house them. Universities can't quite figure out how much to charge us because you can't house them in a mouse cage of 5 animals, which is what most universities work their per diems on, how many cages of mice you have. Our colonies have cages that are interconnected with plexiglass tubing, and my biggest colony at the moment is 200 animals. So they would charge me the same as for 5 mice. So I win on some and I lose on others. But the per diems for them are high. We do all our own feeding and cleaning, and we just pay rent for the rooms, so to speak, as our per diems. Rent, heat, and electricity, I suppose.
This episode of Levity is brought to you by Vitalism Foundation. It's a community of action driven by vitalism, a new moral philosophy whose first tenet is that life and health are good, death is humanity's core problem, and aging its primary agent. They're one of the few groups that truly understand aging as the emergency that it is.
Vitalism's goal is nothing short of monumental: to allocate 1% of global GDP to this project. It's ambitious, but it's exactly what's needed to tackle aging head-on.
As a member of Vitalism, you'll get access to a private community, exclusive content, and invites to special conferences. Plus, you'll receive insider prices on longevity wellness services from Vitalism's mission-aligned partners.
But more than that, you will be supporting a mission that's too important to leave to others to tackle.
You should really join us. Follow the link in the description below. For a limited time, you get 30% off forever. Just use the promo code LEVITY at checkout.
Again, that's 30% off forever with the promo code LEVITY. Let's make a difference together.
Yeah, maybe we should turn to their role in aging biology and why they're so fascinating when it comes to longevity, their incredible resilience. How long do naked mole rats live, and why is that so surprising for an animal their size?
So naked mole rats are the size of a mouse, big mouse, 35 grams. And an animal that size should live about 6 to 8 years. Mice are extremely short-lived for their body size. They live only 3 to 4 years, whereas mole rats live, my oldest animal made it to just over 40 years. And I have many other animals that are in their 30s that are still going strong. So maybe we'll beat that record eventually. So just based on allometry, they are living 5 to 10 times longer than they should on the basis of body size alone. Whereas in every other species, apart from humans and bats, aging and maximum longevity correlate very well with maximum body size. The first reason is they're extremely long-lived as a species. The second reason is when you look at rates of aging and rates of dying, we know from the early work of Benjamin Gompertz in the 1800s, who did the actuarial tables for how much people should be paying for insurance based on their age, that you reach a point at about 3 times the age of sexual maturity where you start doubling your risk of dying. So once humans get to about 35 to 45, they double their risk every 8 years of dying. In the case of rodents, in mice, it usually is about 10 to 12 times the age at sexual maturity, because mice become sexually mature at 1 month. Mole rats become sexually mature at about 6 months, and we've gone to 40 years without seeing this exponential increased risk of dying. So they seem to not show an age-related increased mortality rates. Those are 2 very important reasons why I think they're an important model for aging research.
And in 2018, your team published the first major paper showing that naked mole rats appear to defy, as you said, the Gompertz law. Their risk of death doesn't increase with age. And I mean, that's unheard of when it comes to mammals, right? Almost, anyway, I think. And
I think it's unheard of when it comes to just about every species, never mind mammals. It's true for birds, it's true for reptiles.
And I think that when you published your results in 2018, it came as quite of a big surprise to aging biologists, right? And even skepticism. But then last year in 2024, you published again with more data, and you confirmed and you could strengthen those original findings, right? So tell me about, maybe you should start with the first paper. What was your reaction to that?
Well, I always knew that death was stochastic in my colony. I had as much of a chance of a 2-year-old dying as a 25-year-old dying. So I wasn't totally surprised by the risk of dying. We'd already done studies where we showed that body composition, muscle mass, heart rate, fertility didn't change as the animals aged. And we know from human studies that once you reach the ripe old age of 35, everything is on a downward slippery slope. Your nerve conduction velocity, the speed at which you can run, everything, kidney function, everything starts to decline somewhere between prime 20-year-olds and 35-year-olds. So I knew that they were showing a negligible senescence phenotype in their physiology, which told me they were able to maintain tissue homeostasis and normal function even into old age. I didn't know at that time how long the animals would live. So it seemed like every 5 years I was publishing another paper saying mole rats live to 27. No, mole rats now live to 37. And now my oldest animals made it to 40. And I say, well, I think that's probably as long as they're going to live, but we'll see. I don't know.
What happened to the rats that died? Why did they die?
I wish I knew what they die from. We keep our room really warm, so unless we find an animal in the process of dying, when we do our necropsies, we can't really get much more information than is there a tumor, is their heart enlarged, because their tissues start atrophying pretty quickly. But we have found that we've had very few cases of cancer. We've had, I think, no more than 10 cases that we can document. We've seen one case of a clearly enlarged heart, but we killed the animal, so we don't know if it would die from it. It looked sick at the time. The pathologist reports say most of our animals seem to die with either periodontal disease, sort of abscesses in the mouth and gums, or from renal disease, where the cortex and medulla seem to collapse into each other, and the kidney doesn't seem to have its normal anatomical features. But we haven't really come up with a clear cause of this is the most common cause of age-related death.
But these causes of death, are you saying that the probability that they will be afflicted by them is the same when they are 10, 20, 30, or 40?
It's the same when they're 2 to 40, yeah. So we've had a 2-year-old with a tumor. We've had 2-year-olds with periodontal disease. But my feeling is that periodontal disease is the last thing that happens when you no longer feel like eating because you're sick and dying, and the bacteria in your mouth start eating your gums and cause ulcers. So I don't like to say that they died from periodontal disease. I think it's just a manifestation that something else is killing them.
So do their teeth wear out? Or?
No, they're constantly growing. It's not that they lose their teeth, but when they're digging, the teeth lose their calcium and get smaller and smaller, but they can regenerate teeth throughout their lives.
So then do they lose the teeth and then get a new pair, or?
If they break a tooth, a new tooth will grow in its place. If they've worn their teeth to the gums, the teeth that are below the gums will start growing and get longer.
Yeah, so you published in the first study in 2018 about this, and then in 2024, as I've said, you published again with more data. What was the reaction you got when you published in 2018?
We got mixed reaction, like everything. There are people who are skeptical. We had someone say, your data can't possibly be real because you don't necessarily have monitored data from the beginning of time. You only have a 6-year period in which you've monitored your data of known age animals. And so we had to do a whole lot more actuarial statistics using left censorship and right censorship, and we got the same results in response to that critique. In our 2024 paper, which had obviously many more data, we could use the separate 5-year data set to show that we got almost identical results to what we'd shown in the earlier paper, and then we could combine them to have a much larger period of time of monitoring and we still got the same findings. And intriguingly, we found that the breeding females lived the longest of everybody. And when we did Kaplan-Meier survival curves, we noticed that the median lifespan for subordinates was about 19 years, suggesting that's the time at which 50% of the population had died over that period. If you then expanded that, we'd say that subordinates should live about 38 years, which is about what we're seeing in captivity. 40 years, 38 years. I'm not going to fight over a 2-year period. But when we looked at the breeding females, not only are they reproducing without showing any sign of menopause throughout their long lives, but we're seeing that we haven't got to the 75th percentile. So we don't know what their median lifespan is going to be at this point in time. We can only say from what we've monitored in our population that 35 years isn't the 25th percentile. So does that mean they'll live till 100? I do not know. I'm not going to be around to find out, but certainly they live the longest of everybody. And the next cohort that lives longer than the subordinates are the breeding males. So it seems that being a dominant animal affords some greater cytoprotection, and it might be linked to stress hormones than what you see in the subordinates, but they are still remarkably long-lived.
What do you know about this difference in lifespan between the queen and the subordinates?
Well, from an experimental perspective, although this doesn't obviously affect their lifespan, we don't use the breeding females in our experiments. So we have a larger population of older breeding females and breeding males than we have of subordinates in our colony. But to me, it's fascinating that the breeding female, any female, as I mentioned earlier, can become a breeder. So there's nothing that they have better quality genes or they're a higher quality individual, because I can take out any individual and pair it. Even the animals that were losing the fights become breeders and live a long time.
Right.
So that's not going to be a component of their extreme longevity. What was intriguing to me was that Steve Horvath, who does work on the epigenetic clock, looked at the epigenetic clocks of breeders versus non-breeders and found they had lower DNA methylation. And that to me is fascinating, because that tells me that the DNA methylation clock isn't irreversible. There has to be something that happens that a female, when she becomes a breeder, regardless of age, switches off that DNA methylation and has a more useful phenotype. And I think that would be something well worth trying to understand. How is this reversed?
Is nature's own reprogramming factor at work, maybe?
Yeah. Interestingly, also, when a female becomes the breeding female, regardless of her age, she undergoes a pubescent growth surge. So her lumbar vertebrae lengthen, not the space between them, but the actual vertebrae lengthen. But her legs don't, so she becomes disproportionately longer than the others in the colony.
Yeah.
And this enables her to have much larger litters. So her first couple of litters are between 1 and 9. And once she's a longer female, she can carry more offspring in her uterus horns and can have up to 30 pups in a litter.
Yeah, everything about naked mole-rats is fascinating. They don't just have one party trick, so to say.
No. But I think that's true. If you're going to live a long time, you're only able to live as long as your weakest component. So every organ has to be maintained. And there's been some wonderful work that's shown that another weird feature of breeding females is that they're able to maintain oogenesis into adulthood. So if you think of humans and mice, all our primordial follicles are present at birth, and we use them and then become less fertile as we have fewer eggs that we can apply for breeding. in the case of mole rats, there's been quite a good paper, I think last year in Science, by Miguel Obrino, who showed that oogenesis is maintained and they are able to make new follicles, which would contribute to why they're not going into menopause.
Okay.
And why they appear to have higher fertility in established older females.
One thing that I've always thought of, and now I have the world's top expert to ask, is that if the naked mole rats doesn't age in the usual demographic sense, what do we know about, why don't they live longer than 30 or 40 years? As you said, we might not know that. We might not have that answer right now, but still it seems like there's some sort of limit. So I guess they do age a little bit, or how should I think about that?
Yeah, that's something that I've sort of tried to wrap my head around a few times, because when you look at a population, you're having death at any age cohort coming along. So eventually your oldest cohort is going to run out of lifespan. If only 5% of them die per year or per 10 years, you're going to run out. So I think there is a limit to how long you can maintain stochastic death for a start. But I don't know what their lifespan really is. I would be very surprised if it's much more than 40 years, because that's so unusual already for a mouse-sized rodent.
Yeah.
But I really would be fascinated to know, if I could only get your grandchildren to finish my studies, can they make it to 100 years? Will there be some individuals that I've chipped that should make it to 100 years? Chuck Finch sort of reanalyzed our data from our 2018 paper, and he claimed, based on what we had shown, our oldest animals should make it to 100. I don't know if that's possible.
Yeah.
I personally don't think it is, but I don't know. I'm just a skeptic.
Maybe we should talk a little bit about the different phenotypes and features that helps the naked mole rat to be this almost ageless species. They almost never get cancer. I mean, you mentioned now a tumor for a two-year-old, but that must be incredibly rare.
Yeah, we've done thousands of necropsies in our colony over the years. Every time an animal dies, regardless of how dead it is, I try and open it up and see what I can see. And if I see anything that looks vaguely unusual, I collaborate with the pathologist who has specialized now on naked mole rat pathology at the Brookfield Zoo. So she now has done all our difficult pathology, and has come up with the fact that we've had 10 cases of cancer in about 7,000 necropsies. Compare that with mice, where 70% of mice, when they're 2 years old, have a T-cell lymphoma if they're black 6, or other tumors if they're a different strain of mice. So the cancer incidence is very rare, and is much more rare than what you find in humans. But they still can get cancer. It's not that they're totally invincible to everything. When we look at the types of cancers we've had, the cancers we've had are all over the place. We've had a liver cancer. We've had an esophageal cancer. We've had a salivary gland tumor. We've had mammary gland tumor. And we've had an adenocarcinoma, as well as one T-cell lymphoma. So we know that they can get these cancers, but we very seldom see signs of metastases. Often we've seen massive, the mammary tumor was massive. It was almost as big as the animal, and yet it hadn't metastasized into tissues. The only cancer that had metastasized was the T-cell lymphoma, where we found T-cells in the lung and the kidney and everywhere. But they could be just moving in the blood rather than a true metastases forming. So yes, we've seen a few cases of cancer. we've seen one case of a heart attack where we found scar tissue in the heart tissue, but it was old collagen rather than staining with the stain for newly formed collagen. So it looked like the animal had survived whatever myocardial insult it had had. When we've looked at brain situations, we see very high levels of beta amyloid and tau from as young an age as 3 months all the way to 30 years, but it doesn't increase with age. And we see no signs of plaques and tangles. So they seem to have issues that might lead to disease without getting the disease.
And in the case of cancer, there's something called the hyaluronan. Do I say that right?
Hyaluronan. Hyaluronan is a component of the extracellular matrix. It provides the elasticity of the mole rat tissues. And in cells in culture, if you don't feed them every day, they exude vast amounts of this hyaluronan, which makes the cell culture media very viscous and makes the cells not grow normally. And there's been some indication to suggest that the hyaluronan prevents the cells from anchoring in the cellular matrix, and especially the cancer cells, and so they don't really develop. So it's possible. The mole rat hyaluronan seems to have a higher molecular weight than that of mice, and that might play a role. But I don't think that's the be-all and end-all. I think it's just one very small component.
I mean, we often talk about the hallmarks of aging on this podcast. It has been mentioned a few times, and so if we talk about like proteostasis and genome stability and telomeres and so on, what do we know about the naked mole rats there?
We've got a lot of information that just about every hallmark of aging in mole rats is abrogated and delayed. So their telomeres do shorten as they get older, but they reach a certain level and that activates telomerase, and then they maintain that level for prolonged periods of time. So in a study in cell culture, most cells for mice get to about 18 to 20 population doublings, and then the cells stop proliferating. Hayflick limit for humans was 52. We've got to 160 in the naked mole rats, and that took multiple years, nearly 8 years worth of growing cells in culture, and we don't see that senescence phenotype. And we think it's because they can maintain their telomeres and telomerase. With regard to proteostasis, every aspect of proteostasis seems to be tightly regulated and well maintained as the animals get older. Their protein formation is much slower than what you see in mice, and their protein half-lives seem much longer. So their proteins seem much more stable with lower rates of synthesis. So there's less error or more time to correct error when they're making new proteins. They seem to have very high levels of heat shock proteins or molecular chaperones, which I think contribute to the fact that their proteostasis is so well maintained. They're able to maintain their protein structure more readily. There's high levels of ubiquitin, but the proteasome, the garbage disposer of the cell, seems to be able to work much more efficiently than that of mice. And even autophagy, which gets rid of protein aggregates and mitochondria and things like that, when we do it in culture, the maximum rate is much, much higher than mice. So they can ramp up their machinery quite efficiently to get rid of damaged proteins and well maintain proteostasis. I think that's a very important component of their extreme longevity.
And also their immune function is also playing a part, right? They have an incredible adaptive immune system, right?
Yeah, the immune system really had me flummoxed. I decided mole rats clearly hadn't read the textbooks with regard to how they should behave there. So we looked at it. Our initial study that Hugo Hilton led on single-cell transcriptomics of the spleen showed that naked mole rats, unlike mice, predominantly had myeloid cells, macrophages, and neutrophils rather than lymphoid cells. So they seem to rely much more heavily on on innate immunity rather than adaptive immunity. So when a problem arises, they're at the ready. It's a fast response. It can deal with whatever insult has come their way rather than wait for antibodies to be made and for the adaptive immune response to kick in. Moreover, and intriguingly, they lacked natural killer cells. So we know that natural killer cells are terribly important for identifying viruses and getting rid of viruses, and for immunosurveillance and preventing cancer. So you scratched your head thinking, what's going on? It makes no sense to not have natural killer cells given their cancer resistance. But given their viral response, I think that's their Achilles heel. I think they're protected from viruses because they live underground in a secluded environment protected from airborne pathogens. But when they've been exposed to pathogens, and luckily not in my colony, touch wood, people have lost their entire colony due to a virus. So
Right.
Cornell lost a colony to a virus. Cape Town identified a coronavirus that wiped out their colony, and the only ones that survived were the breeders. So I think they're not very well protected against viruses, but they don't really encounter them, so it's not a big deal. And I think with regard to cancer, they have an unusual population of T cells. They predominantly have in their blood gamma delta T cells, which are usually found resident in your gut to deal with pathogens coming that way. And in human studies, humans with a high population of resident gamma delta T cells have a better prognosis surviving cancer than those that have a lower proportion of them. And yet mole rats have these gamma delta T cells in their circulation, and I think they must play a very important role in dealing with mutated cells, senescent cells, and the likes thereof. So I think the immune system plays a very important role in their cancer resistance and in the removal of damaged proteins and things. But I don't think, again, it's the only answer. I think every aspect of their biology plays an important role. We know that there are a lot of mutations in their proto-oncogenes, so they lack the BRCA gene, and they have mutations in Wnt signaling and MYC signaling, which are all meant to be very important in cancer. And they seem to have positive selection for other genes that protect against diabetes and from the downregulation of mTOR, which is one of the hallmarks of aging. They show signs that they've got mutations there that lead to lower levels of insulin IGF signaling and mTOR secretion.
Yeah. I should correct myself. I said adaptive immune system. Of course, it's the innate immune system that they have that are unusual. Maybe we should phrase it like that. But one thing I'm really curious about as well is that, I mean, they don't have these properties, these phenotypes, because evolution selected them to be long-lived species, right? So why do they have all these protection? What are they there for to begin with?
Yeah, that's a good question. I think all of these protective mechanisms that they show are linked to life in an extreme environment.
Yeah.
So because food is scarce, protein translation and protein synthesis isn't something that they have necessarily needed to constantly turn over. They've been trying to be economical in terms of their energetics and in terms of their making of proteins. I think that plays a role. I think in terms of the kind of environment they live in, they need to have cytoprotective mechanisms against toxins on food, plant cyanides and things on the food. So they seem to, and pesticides in soil and soil heavy metals. And so they've developed an upregulated NRF2 signaling pathway and other cytoprotective signaling pathways to protect against the toxins they would find in their environment. And this stress resistance contributes to helping them deal with the stresses that are associated with aging. So I don't think they evolved to live a long time specifically. I don't think that's what was selected for. I think cytoprotection was selected for, and as a consequence, living in a protected environment where they're not exposed to climatic extremes, pathogens, or even predators, they've been able to positively select longevity mechanisms rather than mechanisms for dealing with those kind of issues.
Yeah, I'm thinking, it feels like there's still a part of the puzzle missing here where I guess you could say that evolution would select for things that would be beneficial for them in that environment, but you could still see another situation where they didn't defy the Gompertz law anyway, right? That they would still thrive, they would still breed, but they would still die at an exponential rate. But they don't. Do we know why that is?
I think living, as I said, as an extremophile in an extreme environment, they don't know if there's going to be no rain for the next 20 years.
Yeah.
And if there'll be new nutrients available. They might not be able, they won't breed if conditions aren't favorable. So they've got to be able, for the species to survive, they have to be able to survive prolonged periods of uncertainty. And I think the harshness of the environment forces that component to be positively selected for. To be frugal in your use, to idle on low, to have a low metabolic rate, a low heart rate, a low set of functioning. But when you need to ramp it up, you can ramp it up immediately. And you see that, especially with their cytoprotective molecules. I cannot understand why an animal needs to have NRF2 levels tenfold higher than what you see in mice when there's no stress. Whereas mice have to encounter the stress, activate the transcription of those pathways, and then deal with the stress. Mole rats seem to have the army primed at the ready for dealing with situations. So you see that with P53, which is the tumor suppressor gene. It's very stable and upregulated even under non-stressed conditions. NRF2, upregulated under non-stressed, unless captivity is a stress condition. Heat shock proteins, everything seems to be at a much higher level under basal conditions than what you see in mice. And why is that the case? So I really do believe that all the cytoprotective mechanisms of naked mole rats are primed to deal with the stress long before the stress arises, because of the kind of environment that they've evolved in, and that this contributes to their longevity. And as such, I think they probably have different mechanisms to deal with the chronic stresses that humans encounter, like the low oxygen when you have a stroke, or the high sugar content when you have diabetes. They have to deal with these things on a daily basis to survive in their natural environment. And if we can find out how they survive them, maybe we can apply those to human health to get better mechanisms for dealing with the same chronic stresses that occur in most pathologies.
Yeah, maybe we should talk about that now. Well, that's something that is people often wonder about. Why don't we find more translational value from naked mole rats? Is it the case that they are harder to maintain or more expensive to maintain, so we can't do as much research on them as we would like? Or you are at the epicenter of this, so you know this better than anyone.
Well, I think over the last 100 years, most biomedical research has consolidated to focus only on 4 species: fruit flies, yeast, worms, and mice. And even amongst mice, only one strain of mice, C57 black 6 mice, is predominantly used. And I think it's easy. We have great experimental tools for using these traditional conventional models. And we've learned a lot from these conventional models. Don't get me wrong. We know that a gene manipulation can extend lifespan. We know that dietary restriction can extend lifespan. But in all those models, none of them have extended lifespan beyond what their predicted maximum lifespan should be. So they've enabled them to catch up, but not extend beyond that. And I think because of those experimental tools and the ease of use, people haven't experimented with species that are out of the box. Remember, there are 8 million species out there, and there are lots of animals that we just don't know anything about their biology. And they are a wonderful, untapped resource that I think can help biomedical research, especially the animals that live in extreme environments, be it hibernating mammals. How do they survive with muscle atrophy? Why don't bears get muscle atrophy? You break a leg, your muscle deteriorates very rapidly. If you think of animals that are flying at high altitude, or seals and whales and all these species, many of them are very hard to keep in captivity. So I understand, how we're going to keep a blue whale in the lab. It's not going to be easy. You might be able to do stuff with their cells, but you couldn't do stuff with the whole animal. And I think because of that, the convenience of conventional model systems has really constrained biomedical research. And I think there's tremendous opportunity for scientists to think out the box and start working with non-conventional species. And drugs are being made from non-conventional species. I'm sure you've seen the Ozempic drug, which was derived from the Gila monster saliva, or the ACE inhibitors that came from the venom from a Yarraka pit viper venom. So, there are things that are out there. It's just that it's harder to do that kind of research.
Can I ask you, how many labs are there focusing on the naked mole rats? How many researchers are engaged in this?
Well, up until I published in 2005 that the naked mole rat was an exceptional model of biogerontological interest, there were 3 or 4 labs. And now there are 125 that I know of. And many more scientists are collaborating with those of us who have mole rats to get animals or tissues to conduct their studies. So I think the interest has grown. I think the naked mole rat has made its way from being an esoteric, idiosyncratic, crazy animal into the normal mainstream use in biomedical research, especially in cancer and hypoxia research.
I mean, it's a model organism in a different way than other model organisms, because it's a model of how we want to age. Not at all.
Absolutely. I do believe it holds the blueprint for successful aging. If only we could figure out all the components that make it up. We've uncovered several, but I don't think it's anywhere near the whole story.
I mean, I think the rational response to knowing this about the naked mole rat is that we want to know everything we can about it as soon as possible.
Yeah, and I think that's been the problem with drug companies. You can't run a marathon if you're still learning to crawl. And the drug companies want a drug now. They don't want to wait for you to understand the basic biology and physiology of a species and be able to then tap into the differences that might be useful for human therapeutics.
But do you consider the area woefully underfunded, or how many labs should there be?
The area is woefully underfunded. It's seen as not going to yield results quickly enough for commercial application. And because of that, I think we've got real problems in terms of exploiting the many species that are out there for such research.
So basically, we found the fountain of youth in this animal, but we're not sure that we can sell anything very quickly of it.
I think that's part of the problem. But, there are people, especially in my lab at the moment, we're collaborating with Argon computers to try and find small molecules that mimic or recapitulate mutations that we're seeing in different pathways. And we're trying to see if these molecules can then induce the same kind of functional responses that we see in the mole rats. So there is research that's working in that area. But as I said, first you have to understand the problem, then you have to work out what are the solved mechanisms that the animals have used, and then try and mimic it using some kind of drug.
Besides small molecules, are there other translational avenues to look at as well?
I think finding a drug is the ultimate goal. I don't think we want to mimic the lifestyle of the naked mole rat. So I think they, if we can find out how they have managed to reverse their methylation clock, I think that would be an important finding. We know that they activate telomerase when their telomeres are too short, but we know that studies where telomerase has been activated in mice and other models has just led to cancer.
Yeah.
So it's a double-edged sword. You've got to be very careful in how you do it and how you apply it to medical work.
And I was also thinking about gene editing. That's an area that's moving ahead very quickly now.
Oh yeah, and I'm surprised I didn't tell you that. Yes, CRISPR and gene editing is a very big deal in terms of putting in different kinds of mutations. In fact, we've done that in some mice at the moment and some cells in culture, looking predominantly at the immune system and also at their IGF, trying to get a good understanding of how they've managed to reduce insulin IGF signaling and through mutations, and can we mimic it?
What have you found there?
It's so early in the stages, we literally are waiting for the animals to come in.
Then we have reason to chat with you again in the future.
Yeah, 5 years from now.
And then we will have even older naked mole rats, hopefully.
Yeah, hopefully.
As a comparative biologist, what other long-lived species are you interested in?
What am I personally interested in?
Yeah.
I'm very interested in the bats.
Yeah.
I think they're another very interesting model organism that does things totally differently. We know that their immune system is much more attuned to dealing with viruses and to coping with those conditions. They show many similarities in terms of a very low resting metabolism and heat tolerance and thermolability to what you see in mole rats. And they might be employing similar mechanisms there that might be directly pertinent to aging. What worries me a little bit with my interest in bat studies is hibernation is really a suspended state of animation. So can we sort of think that a bat that hibernates for 9 months of the year and lives 40 years is doing the same thing as a mole rat that is active throughout that period of time? But I think there are interesting parallels and interesting divergences there that might help us figure out better ways for retarding and abrogating aging. I haven't worked with whales or any other of the green sharks or any of the other long-lived species like that, but I think there are many answers that could come from that kind of study.
Yeah, I know that Stephen Ostad has raised this as well. By the way, I wrote about a company called Fauna Bio. Do you know about them?
I know Fauna Bio quite well, yeah.
Yeah, okay. Maybe you've been advising them? I didn't know that.
I know that we've interacted. They're working on 13-lined ground squirrels and hibernation and looking for drugs that might come from those hibernating squirrels. So I've met with them and we've had discussions about mole rats and squirrels and the likes thereof.
Right. Yeah, I think that they said that, I mean, it's difficult to, I mean, it's difficult to work with naked mole rats.
But I'd like to take you to task on the fact that naked mole rats aren't that difficult to work with.
Yeah.
I think before I left Calico, I gave away 7,000 animals to labs in Japan, Korea, Germany, England, France. The list is endless. And they're all doing very interesting research with them. You know, once you've figured out how to house them, which isn't difficult, I've written a whole chapter in a book how to do that and have advised and consulted. I think you can get some very interesting answers to areas where you have specific expertise, and I think people are doing that.
What is the community of mole rat experts like? Do you have mole rat focused conferences, and do you know each other?
I don't know all of them. I know a lot of them. We had a conference this last year in Bonn on mole rats, not just the naked mole rat, but on mole rats. And many of the people working on mole rats were present at that conference. And it was good to talk to them because several of us are doing very similar kinds of research. So, for example, I did a study where we tried to induce brain strokes in naked mole rats and mice and myocardial infarctions in mice and mole rats. We found that their cardiomyocytes could regenerate and that they didn't go into a brain stroke. And while I was talking at the conference, I discovered 3 other groups were trying to do the same thing now. So, you know, I think it's great in that, one, you're getting reproducible results from different labs in different parts of the world. And 2, we all come at the question with different expertise. And so the way in which we ask the question will elucidate different things. So I think, you know, we're just at the beginning of a wonderful new model being exploited in biomedical research. And I think it's going to give us some very important information.
Is there a journal focusing on this research?
No.
Peter?
We publish mainly in geroscience and aging
Peter.
journals.
You think I should start a naked mole rat
I
journal?
think so too, because it makes me nervous that we're not doing all we can to get to know this little creature as much as we can. And we need to do it like as fast as possible.
Yeah, but at the same time, what Rochelle tells us is that it's kind of hopeful, because I think the field is actually bigger than I thought starting this interview with you. I didn't know that there were so many labs around the world working on this. So that's really encouraging.
No, I think it is encouraging. I mean, in 2000 there were 3 labs, as I said, and now there's just more than 100 and growing.
Is it widely accepted now that the mole rat is non-aging, so to speak, or are there still skeptics out there?
I think it is widely accepted. I think every lab that starts doing some study to see if their particular phenotype changes with age, comes back and says, no, it doesn't. So I've been reviewing a bunch of grants which tell me that people are looking at kidney function, and that's not changing with age. And that's one of the areas I have no expertise in and haven't done. So I think the more people who work in the field, the more it'll become widely accepted that this is a real phenomenon, that they're very well able to maintain tissue homeostasis of just about every organ.
And you were, as you mentioned, at Calico for, I don't know how long you were there, quite a few years, I think.
I was there for just under 7 years, 7 years.
I know that's a secretive company, but what did you do at your? Can you say anything about your time at Calico?
Between
what
us?
you did there?
Yeah, my time at Calico was very, very exciting. I thoroughly enjoyed working with people with expertise in areas that I have no expertise in. For example, the demographic aging work was done by the actuarial scientist there. I collected the data and he did all the fancy analysis for it. So that was an important paper to really prove the point that they are a good model for aging research. We also did, while I was there, a platinum genome of the naked mole rat at chromosome level specificity that has been annotated, and that we were hoping to really interrogate to find all the different mutations that might be pertinent to their extreme longevity. And we got that study pretty much underway. We used single-cell transcriptomics while it was still in its infancy to do the immune system function. And we did a lot of work on proteostasis and making of different molecules to sort of look at the interrelationship between heat stress and proteostasis in the mole rats, given their unusual thermolability. So we were working on in multiple directions, as you can see. We also did a study where we looked at skin cancer formation using UV radiation as well as anthracyclines that damage skin and are meant to be pro-carcinogenic. And we looked at the protein changes in there. So we did a lot of basic science. I was there when Calico first formed. I was in the first cohort. And most of the work we were doing was understanding the basic biology of aging to try and go to the next step.
Yeah.
And we left before the next step started, unfortunately, so we didn't get to making drugs. But I think other labs now are beginning to do that. I think Altos Labs is working on naked mole rats at the moment, and they might come up with the first drug for aging.
Is that something you've thought of yourself to spin out a biotech company around this?
Yes, my current postdoc and I have been talking a lot about some of the molecules we're looking at and thinking about the possibility of biotech based on it.
That's another reason we have to have you back then, when you started your biotech company.
Yeah, I just hope I live long enough to finish all the studies we've started. There's so many interesting things that need to do.
I know that Wired wrote a profile on you and naked mole rats a few years back, and they called the naked mole rat Yo. And they admitted later in the article that it wasn't actually named Joe. They just gave it a name. Do you have any names for your naked mole rats, and do you get attached to them in any way? We've just established that you don't eat them, not yet anyway.
No, when I run out of grant money, I might have to, right? No, I personally don't like naming my animals because then I get too attached to them and I can't do experiments on them.
Yeah.
And I think we're at the stage where we need to get these experiments done. I keep thinking of our brain stroke study and all the components of it, which I haven't told you much about, but I can give you a brief rundown there.
Yeah.
I keep thinking, thank goodness I hadn't named the animals, because the whole idea was to give them the stroke and have them run around in circles and see if they can survive it. And we ligated the carotid artery, so we reduced blood to half the brain. And we did the same thing in mice and mole rats. At first, we put them on 8% oxygen, which is the concentration that all the mice got strokes and many of them died. That didn't seem to do anything to the mole rats, so we doubled the amount of time. Then we halved the amount of oxygen, and even then we didn't see a single stroke that we could monitor to see how they recovered. So it was interesting that they were so tolerant of this pretty strict procedure. And I kept thinking, if I'd named my animals, I wouldn't have been able to do that study.
Right.
So I don't name any of my animals. I do get attached to them, though. When the oldest animal looked pretty sick, he was 21 years at the time, and the vet told me I needed to euthanize him because he'd gone very thin. I went in the lab to euthanize him, and I couldn't bring myself to do it. And luckily for me, I then moved to Texas pretty soon after that and took him and everybody else with me. And he lived another 8 years without dying from whatever it was that had made him lose muscle mass. So I don't follow orders when they look like they're ill, and I'm pleased that at least at Calico for the demographic study, we didn't need to euthanize any animal on a vet's command, because the animals seemed to recover from whatever malady they were suffering from at that moment.
I was thinking about whether the older rats, they pass the cognitive tests just as well as the younger ones.
Yeah, they manage to manipulate their way through the burrow system to the food. They find that high protein food fastest and they eat it first. And I'm hoping that our cognitive tests will show that. So it'll be interesting. Our current research is looking at brain aging and Alzheimer's and Parkinson's. We're trying to induce Parkinson's in our animals, given the fact that they have such high levels of the proteins that are meant to be causally linked to these diseases. And we'll see if we can either induce the disease or what the mechanisms are that prevent them from getting them.
Yeah, you mentioned that they have plenty of amyloid and tau, but also that they are very good at maintaining proteostasis. How do we reconcile those two facts?
So I think tau is essential for microtubule formation and for neuron stability, at least in fetuses and in early life. And I think beta amyloid gets a bad rap. I have a feeling that it's a cytoprotective molecule.
Mm-hmm.
So I'm going out the box trying to understand why this is the case. And we see very high levels that are maintained throughout their life. I think what happens in Alzheimer's disease, and it's just a hypothesis, is that whatever's causing Alzheimer's disease initiates a cytoprotective response in which beta amyloid is upregulated, and the patient is overwhelmed when they have Alzheimer's disease, and that's why the beta amyloid is present with the plaques and tangles that are being formed. And I think that in the case of the naked mole rat, the cytoprotection is there, but it doesn't get overwhelmed. You don't see the beta amyloid forming plaques or congregating in the axonal heads of neurons. With regard to tau, it's the same thing. The tau protein is maintained in the mossy fibers, and so it seems to be maintaining its normal function. And I think in the case of naked mole rats, we see lots of evidence of new neuron formation, neurogenesis, even in animals in their 20s, which is the oldest age group we looked at in terms of the brain. And I think that the reason we see such high levels of tau is that they are maintaining neurogenesis and they need tau for their normal neuron formation and structure.
Oh, that's so interesting.
So again, I keep telling you, the mole rats haven't read the textbooks, so they do things differently to other
We're
species.
very grateful for that. That they're doing things the right way. What advice would you give to a young scientist that sees this and gets really interested in how do I work with these amazing creatures?
My advice to a young scientist, firstly, is that aging biology is the be-all and end-all. It's a very exciting field. For many years, it's never been given the same kind of status as chronic diseases or lifestyle diseases like diabetes or cancer or neurodegeneration. It's always been regarded as a kind of fluffy science, and I think that's changed. I think the realization now is that aging is the cause or causally linked to just about all those chronic diseases. And if we can find ways of abrogating or retarding the aging process, we will automatically find ways of reducing the incidence of cancer, diabetes, neurodegeneration, etc., etc. So I think it's a very exciting field to be in. I also think that while it was difficult in the old days when we were dependent on established tools like antibodies and flow cytometry that needed markers that worked in mice, we now have advanced tools that don't need those kind of well-established criteria that kept mole rats and other exotic animals out of the research dimension. And I think with all the advances in the omics and the post-translational modifications, the world is their oyster. I think there are 8 million species out there, and if they can combine a little bit of field work and a lot of laboratory work, they might find novel ways of studying aging using exotic species rather than the more conventional, traditional species. So I think we're at an exciting turn in aging research with all these advances where comparative biology will likely play an important role. And I would say to them, don't give up just because you can't use the tools that have been developed for mice in the 8 million papers that are out there on C57 Black 6. There are new ways of dealing with things. You have to think out the box. But the cost of doing it is exceeded by the potential from what you will find.
Thank you, Rochelle. I think, Patrick, do you feel like before we ask Rochelle to recommend some books, is there something we didn't ask her that you're curious about?
No, it's been very rich, and I hope we have opportunity to speak.
We've been
again.
talking about doing the naked mole rat episode since we started a year ago, so I'm so happy that we finally got around to do it. So, Rochelle, we always ask our guests about 3 book recommendations. Have you thought of any books that you would like to recommend?
Yeah, there's a very good book by Barbara Nuttison Horvitz called Zubiquity. I don't know if you've read that. She's a cardiologist who has been trying to understand why animals faint when predators come and the role of hypertension and like thereof. And she's written a lovely, very easy read on the use of animals in research. So I would highly recommend Zubiquity. I thoroughly enjoyed reading it, and it sort of made me think about different kinds of projects I could do, even though it's a book for lay people. It's not really a book for research.
Yeah.
I also loved Steve Ousted's book, Methuselah's Zoo. I don't know if you read that. That came out, I think, a couple of years
Yeah,
ago.
I have read it actually, and we have other guests as well that have recommended his book, and we met Stephen at the ARDD conference in Copenhagen
Okay.
last year, so we're probably going to invite him to the podcast as well at some point.
Yeah, no, he's a great guy, and his book is a very interesting read.
Yeah,
So I would recommend that one. And then there are a whole bunch of books on Eternity Soup, which was an interesting book looking at aging research across the ages, that I would recommend as well. And The Death of Death, which I thought was also, I mean, there's so many new books coming out in the field of aging and dying that I think people who are interested in aging research will find those books particularly intriguing.
yeah. We had Jose Cordeiro, who wrote the Death of Death on
Yeah.
the podcast recently. Yeah, he will be very happy that you recommended this book, I'm sure.
Yeah, no, I thought it was good. And I'll turn the question back to you. I think I need your book and your book.
Yeah.
I need to start reading new books as well, but
Yeah,
those are the ones that immediately come to mind.
Mine's only in Swedish so far, but I can point you to my newsletter. But Patrick's book, The Case Against Death, you should definitely read it. It's great. And I don't
will
just
order
say that
it on
because
Amazon
he's the
today.
Excellent. Yeah, I'm discussing all the arguments people have made through the times in defense of aging and death.
Okay, I will definitely read it.
And
And
I
of
was
course,
not
if people are
persuaded by their arguments. I think I'd rather be youthful and alive.
Yeah, I think I might agree with you. And of course, there's my book, The Extraordinary Biology of the Naked Mole Rat.
Exactly. I'm glad you brought that up. Where can one buy that? Do you find that on Amazon, or where do you find
You can
your
get
book?
it on Amazon, but you can buy each chapter individually, or you can get it through your university library, because it was through the Advanced Biomedical Sciences program that we published it.
Perfect. And we'll link to that in the show notes as well. Rochelle, I think between the lines here, if we haven't established it already, I mean, you have brought this field of studying naked mole rats into the forefront of aging biology. I think it's fair to say, and so you've done an extremely important job. We're so happy that we got to talk to you today. So I can just say thank you, and for all the knowledge you brought us today as well. I hope our listeners and viewers will like this episode.
I hope so too. And if you have any questions, feel free to email me.
Perfect. Thank you so much.
Thank you. Thank you for the opportunity.